it's been a fascinating meeting so far. So let's talk about how we can take some of the ideas that have been proposed and think about novel clinical trials for LGS. So the roadmap is, I want to start by highlighting some differences and maybe some similarities between LGS and other early life epilepsies that have had some success recently, clinically or from a regulatory fashion or both. Then we'll get into the details of what an FDA-endorsed clinical trial looks like. And then we'll talk about some novel trial opportunities, some of which have been discussed, but I will try to build on what's been said when it fits. So there's been a lot of attention, deservedly so, recently, about advances in the treatment of tuberous sclerosis complex, infantile spasms, and Dravet syndrome. And these are important for LGS, but I think we have to appreciate the differences. So let's talk about early diagnosis first. So for TSC, that can be done often prenatally, given the cardiac findings, or a heart murmur on exam in the newborn period, sometimes a skin exam. So sometimes a very early diagnosis is possible with tuberous sclerosis. Infantile spasms, with careful tracking, you can maybe determine the onset of clinical spasms within a few days and then quickly verify it. And Dravet syndrome has a typical enough phenotype and rapidly turnaround genetic testing that within a few weeks, you can get a confirmed genetic diagnosis. At this point, I'm not sure any of those are true for LGS in terms of the rapidity of early diagnosis. Etiology for TSC, it is, a genetic mutation. There are, two different genes and multiple mutations resulting in both genetic and structural abnormalities. IS is a varied and vast list of etiologies, and Dravet syndrome, at least SCN1A positive Dravet syndrome, is a mutations in a specific gene. LGS fits in the varied and vast category like infantile spasms. EEG for TS, the EEG is variable, especially at onset. Infantile spasms, you heard about hypsterythmia. It is characteristic, although inter-rater reliability can be challenging without careful definitions. And Dravet syndrome, the EEG is all over the map. You've heard about slow spike and wave in GPFA. It is characteristic, but it's not always there, and it can go away with age. Pre-seizure treatment, maybe it's coming soon with TS. Dr. Grinspan had some interesting data about pre-treatment of infantile spasms beyond TSC, maybe. Early treatment of Dravet is challenging because you need a newborn screen showing the mutation, and you need detailed understanding of the of that mutation's phenotype to do pre-treatment. So pre-seizure treatment is a challenge for everybody, including LGS. What about disease mechanisms? Have they aided novel therapies? For TSC, I think the answer is yes. For spasms, I'd say no. And for Dravet, for the precision investigational therapies that are still investigational, yes. But the approved treatments for Dravet probably work or have been proven to work for other epilepsies as well. So the unique mechanism of Dravet syndrome maybe hasn't informed our approved treatments yet. Bottom line, how many FDA-approved treatments do we have for these conditions? For TSC, it's 2. For spasms, it's 2. Prednisone doesn't count as an FDA-approved treatment. And Dravet syndrome is 3. So a lot going on. And obviously, LGS would like to take the best of this group, take the best, most favorable features to move things along. But how does LGS stand up to these same categories? Well, early diagnosis is certainly an important goal, but it's challenging. And pre-LGS, I think, is still a novel concept certainly to me and to most of us. And there's a lot of work involved in unpacking pre-LGS. We talked about the etiologies, varied and vast for LGS, just like for spasms. The EEG, Dr. Worrell this morning reviewed the EEG. Yes, slow spike and wave is there. I agree with Dr. Grinspan that inter-rater reliability will be difficult, and then it's not there, and then it goes away, and then GPFA comes, but it's not always there. And is GPFA ictal or interictal? A lot of hard work to do with the EEG markers of LGS. We've talked about the challenges with pre-seizure treatment. Disease mechanisms aiding novel therapy in LGS. Well, not yet, but I've been listening today, and I sure hope that some advance can turn into a therapy based on better understanding of disease mechanisms. Bottom line, LGS is winning the FDA-approved medication battle with 6 approved treatments, likely more coming, and that doesn't count valproate, which I think we'd agree is effective, even though it doesn't have an FDA approval. But as Dr. Berg said this morning, these FDA approvals have not transformed the condition in any way. Some patients have fewer seizures and hopefully better side effect profiles, but lots of work to be done. So how do we do this work in the confines of an FDA-endorsed clinical trial? So these are some of the factors to be considered in a clinical trial. Obviously, safety and ethics come first. Inclusion criteria, I think there's room for some novelty. The LGS trials to date have not been new or recent onset. They've been quite treatment-resistant patients, and they've been broad. Maybe there is a role for some specific LGS trials, like the EPGP cohort, that patients like that Dr. Patel presented this morning, patients with, quote, cryptogenic LGS. You leave a lot of your patients behind if you do that, or you could get precise. LGS secondary to SCN2A mutations. That would be precise. You'd leave a lot of patients behind. The more precise you get, the more and more patients are not eligible. Of course, for a novel trial, you need a treatment. It could be a drug, it could be a device. And then there's the issue of the control group, which we'll talk about. Trials can only be so long, especially if you have a concurrent control group that might be a placebo. Usually 3 months is your maximum. And then, of course, you need outcome measures, and Dr. Berg gave an excellent review of the troubles with most outcome measures for rare encephalopathic epilepsy trials. So the other thing about the FDA is you have to show you are better than something. The FDA, for regulatory approvals, wants to see superiority. They do not want to see non-inferiority or equivalence. You have to show you're better than your comparison group. In Europe, it's a little more flexible because a non-inferiority study, if you show non-inferiority, that can lead to an approval. But I'm going to focus on the challenge of a superiority clinical trial. All right, so let's think novel here. Let's be bold. Here are the tough issues with novel clinical trials. Let's say we're really good at getting early clinical onset or early treatment resistance. Is that really when the disease begins? You probably need to understand the biological onset of the condition, not when did the seizure start or even when the slow spike and wave first appeared, but what was the biological onset of the condition? That's a tough one. Something to think about if you're thinking about novel trials is what is the age of the earliest possible enrollment in a randomized controlled trial? How young could you go? Because if we're looking for disease modification, early is probably better, although we don't know that. So biological onset is a challenge. The trial design in the US for now shows superiority versus a parallel control group. And typically you have a max of 3 months to do that before your control group will lose steam and just won't enroll. So you have to quickly show superiority. We can do it for seizures. We have to figure out how to do it for other things. And then, of course, the outcome measures, seizures, development, EEG, and other biomarkers. We'll come back to that in a little bit. So these are tough issues if we want to get novel. And then disease modification. Now, Tracy started off the day with the definition of disease modification, which is reasonable. The FDA takes disease modification seriously. And here are 2 trial designs that would support evidence of disease modification. So the typical RCT is complicated enough. Imagine one of these, a delayed start design. One subject group starts on treatment later than another. And the failure to catch up with the first indicates that there's been an enduring effect on the disease. So the failure to catch up, your earlier subjects do better and they stay better. Or sort of the opposite of that, the randomized withdrawal design. A treated subject is withdrawn from therapy, but they don't assume the same baseline untreated level. So you take away the therapy and they still do better. These are high bars, but if you really want to talk about disease modification from a regulatory standpoint, these definitions are worth some thought. All right, so how can we get novel with LGS? Some of this has been covered today, so I won't duplicate it. I think there is work to do with earlier diagnosis of LGS and perhaps earlier enrollment, even in a traditional LGS RCT. I think Dr. Berg presented very persuasive data that our 6 approved treatments, none of them have been transformative. So if you have a diagnosis of LGS, do you really need to have failed 4 or 5 or 6 therapies? So consider earlier enrollment in the even traditional LGS regulatory trials. Who's in the studies? So far, it's been all comers. LGS broadly defined with all etiologies have given us our 6 FDA approvals and maybe more to come. But maybe if we keep doing that, we'll get more of the same. Another treatment with an FDA approval that won't be transformative. So I talked about one example of splitting, doing a subgroup of LGS, the no etiology found and some very typical characteristics, or some genetic subgroup that develops LGS. But know that this can be tough on the community, these subgroup studies or precise trials, because most of your families will be left behind, and that is difficult. The control group, just difficult. We'll come back to that in 1 second. Let me go back to the precision, for example, as an example. So the poster child of precision therapy in pediatric epilepsy belongs to Dravet syndrome right now. This study is in progress, the safety dosing arm. It's not an efficacy study yet. So to be in the ASO study in children and adolescents with Dravet syndrome, you must have Dravet. You must have a pathogenic mutation leading to haploinsufficiency. And this is an example of precision therapeutics. There are patients left behind. If you don't have an SCN1A mutation, you need not apply. The mutation must lead to suspected haploinsufficiency, or you need not apply. So we are starting to build a small resume of precision therapy in epilepsy. This trial came out of a deep understanding of the biology of Dravet syndrome, and LGS has some work to do on that front. The control group, let's say it's a placebo. Everybody wants the placebo to be as short as possible. I have 3 suggestions for shortening the placebo control arm. This, which has been presented before, has 2 features. It has a variable baseline and a time to event outcome. Basically, rather than being trapped in a 1-month baseline, your baseline varies depends on how many seizures you have. So the highest seizure group could have a baseline as short as 7 days, and then you're randomized. And then you stay in either the investigational arm or the control arm until you have the same number of seizures you had during your baseline, whether it was 7, 14, or 28 days. So this can shorten the baseline phase, shorten the placebo exposure phase, unless you're doing really well, and that, in which case, great. There are problems with this design, but it could be done. And this design has been replicated post hoc in a fenfluramine Dravet trial with the same with the same positive result, but it has not been done from start to finish in this design. Another is what's called a blind start design, which has been done in other childhood diseases, but not in epilepsy, as far as I know. So imagine 4 groups, and you each start at a different time. So a quarter of the patients have no placebo arm because they start on active treatment right away. Some people have a shorter placebo, a middle one, or a longer one. So this can overall minimize the placebo arm, and is an interesting design to think about. Another is a platform trial, which would be very doable for LGS, but just takes a lot of cooperation between study sponsors. You have multiple active arms that share a placebo arm. Doable, but a lot of logistics involved and a lot of collaboration involved. And then, of course, the Iceberg Slide to introduce non-seizure outcomes, which Dr. Berg talked about in detail. Obviously, epilepsy are much more than seizures, and any outcome measure, whether it's seizures or a developmental outcome or behavior or anything, the outcome has to be described clearly and accurately, and you have to be able to measure it. And that may seem obvious, but we know sort of good enough, I guess, how to measure seizures. How to measure the non-seizure outcomes can be challenging, and you reduce your eligible participants because if they have to have seizures, then they have to have something else as well. Maybe they don't have to have seizures. That would be an extra novel way to approach it. Back to the control arm and how long you can have a control arm. We have shown time and time again that you can show a reduction in seizures over a 3-month period in a randomized trial. How long will it take to show improvement in a non-seizure outcome? We don't know. Seizures might respond more quickly than non-seizure outcomes. If your non-seizure outcome will take a longer time to show improvement, then you have a longer time in the control group, and that's a problem. But it will have to be figured out based on the intervention and what your outcome measure is. And anything we use for an outcome measure has to be valid, reliable, and easy to use. So Dr. Berg helped me out with this slide. I can skip the whole part at the top because these would be what she called the boutique of outcome measures. And I agree, most of them are not off the rack applicable to LGS. So, disease-specific outcome measures. It can be done. Rett syndrome did it. Angelman syndrome has done it. NCL has done it. It can be done. It is years, probably decades of work, and the challenge with the disease-specific scale is heterogeneity between subjects and variability within subjects. And I do think LGS has lots of both. Heterogeneity between subjects and variability within subjects. So I agree that disease-specific scales can be important, but they are easier said than done. So of course we want non-seizure outcome measures. Why haven't we done it already? Because it's hard. Believe me, it has been discussed for years. It is hard to develop meaningful outcome measures. You have to have a detailed understanding of the epilepsy type, the intervention, and your outcome measure. You're stuck with a short-term clinical trial for now. And we do need input and flexibility from stakeholders, but we just need more of all of these: discussion, research, and pilot testing to think about non-seizure outcomes in LGS. Which brings me to EEG, and I can be quick here because Dr. Greenspan did the heavy lifting. So this is 3 Hz spike and wave of childhood absence epilepsy. It is very amenable to clinical treatment and as a clinical trial endpoint. It is very homogeneous across subjects. The inter-rater reliability for untreated childhood absence EEG is extremely high, on the order of 99% from the NIH trial, and it can go away with medicine. So this is a dream, the 3 Hz spike and wave of childhood absence epilepsy. That said, we have no contemporary regulatory trials that have used this. We have an NIH academic trial, but not a regulatory trial that has used the 3 Hz spike and wave of childhood absence epilepsy as an outcome. Hypsterythmia has been used in outcomes of regulatory trials. It has led to FDA approvals for both ACTH and vigabatrin. Hypsterythmia is complicated, but with careful rules and excluding patients who don't have hypsterythmia by whatever your rules are, hypsterythmia can be a useful outcome measure in a trial. ESUS. This patient is falling asleep here. They fall asleep and their EEG becomes continuous. This is the next page, and let's just say all night long, that's what it looked like. That's pretty striking. There is a trial underway for a investigational compound looking at ESUS. It is unknown, unproven as an outcome measure in regulatory trials. There will probably be some bumps in the road with figuring out how to use ESUS as an EEG outcome measure. And then there is slow spike and wave. So this is the classic textbook example of 2 Hz generalized slow spike and wave. I will tell you this is an interictal pattern, although you could argue with me and say, how do you know that? This could be an evolving seizure in progress. But in this patient, this was interictal. Yeah, I think Dr. Greenspan covered well the challenges of slow spike and wave as an EEG, as a trial outcome measure. The other thing I'd mention about EEG outcome measures is the side effects of the treatments. You know, the reason why a childhood absence epilepsy can be, in at least half the patients, can be well treated is we can treat the seizures, normalize the EEG with acceptable side effects in half of the patients. We don't, even if we can normalize slow spike and wave, what will the side effects be? What will it take to do that? We don't know, but we do need to explore that. But any challenge you have with hypsterythmia or even ESUS, I think you will at least have the same number of challenges with using slow spike and wave as an EEG outcome measure in LGS trials. All right, so that's it. This is the summary, and we have lots to talk about because there are lots of needs and lots of opportunity for novelty. So thanks to all of the people who have helped my thoughts on this topic, and let's get to questions.
Just a quick question, Dennis. That was a great presentation. You know, knowing what you know and all you went through, I keep coming back to some of those outcome measures. So my question to you is, you mentioned it's decades work, but is that something you would recommend that we should try to do a better job? It may take us time, but do we need to get better and start now as far as folks that do research in LGS?
Hard question, and I think others can comment as well. I think you know, the NCL example is a result of a horrifically tragic degenerative disease. You know, NCL, the heterogeneity of the homogeneity of the decline of NCL, where they stop walking and they stop talking, made that outcome measure possible. That's a horrible disease. LGS is a very, very challenging disease, but it is more heterogeneous than NCL. So I don't think that serves as a model. And then you get into Rett syndrome and Angelman syndrome, which are also have varied measures of severity. And I think I'm gonna say I'll ask if anybody else has insight on the development of those scales and their use in trials and whether LGS should go down that same path. I will say I still wonder that LGS is more heterogeneous than both Rett and Angelman syndrome. That's just off the cuff comment, but that's my thought, which means it would be more challenging for LGS to do their own disease specific trial.
Yeah, I mean, just you know, Rett had many years of a natural history study and clinical data collection to understand the spectrum of symptoms in Rett syndrome. And even with that, right, we still are learning more. You I think as Zach alluded to, and this morning was alluded to, there's what is the full spectrum of LGS? What is all the other features? What percent have autism? What percent have different degrees of intellectual disability, right? So I still think there needs to be some clinical, further clinical understanding of the condition. And where LGS gets complicated, as we've all mentioned, right, is there's many different etiologies. So how do you deal with the many different etiologies? But I think, there's a lot of talk within Rett syndrome and other disorders, right, that there needs, and I think Anne brought this up earlier, there can be some shared things here, where like the new ORCA measure for Angelman's that there's a lot of discussion about for a communication measure, which is a low-end communication measure, that could very well work for Angelman's. The Rett clinical severity scale that Jeff Newell is adapting, right? It has features that could potentially work for LGS, right? So I think there needs to be ways to try to share these things and learn from all the others before just going directly to build one for LGS. I think we'd be losing a lot of learning that we've had over the years if we just dive right into a new one for LGS without taking from all these other disorders, but still trying to get the kind of what is the core of LGS that we don't fully know yet.